little white thing when I
Oh,
get
there's
over
a
there.
nice,
Yep, it's a pen.
which one? Is that a rock pen? The one near us or far away?
That's a black coral, I think.
See if I can stabilize on this one.
Is that
Holding.
Pathies or something?
You might be onto something there, Ed.
Okay, go ahead, Ed.
Coming in.
I don't know. I don't. The skeleton isn't black, but it's shaped like a. It's shaped like a black coral.
I got another bunch of zoom.
I don't know if I can. Maybe the skeleton is black. I can't tell.
It's all my zoom.
Gonna go ahead and pull my not a biologist card on this one.
Yeah, I'll say.
It does not look like a
I'm gonna risk a tip down here. Okay.
We're getting a vote for Bathypathes.
Vote.
Pathies, cool.
I don't know. There's not, somebody who knows more than me.
Yeah. Black coral.
Okay, if we're not sampling, I'd like to move along.
Yeah.
Okay.
Ship is holding,
Roger
by
that.
the way.
I see Babiargus is cruising around down there.
Yeah, I was down there the other day giving the new video team a tour of the space. And I came up here and we looked at the monitor and it was back and it was not down there when I was down there.
What was not down there?
Babiargus' hand.
Oh yeah, it's just been put back. I don't know who put it back, but
It's also at a creepier angle, if I'm honest.
yeah.
Now it's kind of like coming at you.
Yeah.
It's shorter fingers.
Yeah. I like it less scary.
Yeah, Ed, I meant actually Babiargus. There was a winch tour just now.
We might want to come up a bit there on the winch. Get that wall coming.
Look at that traction lower slack.
Haven't seen that.
Hasn't snapped yet, actually.
No.
Let's see the snap.
Have we gotten an official word on if that's all right?
Nothing official.
It just is.
It just is what we're doing.
Well, Emil, you nailed it. This is where we're going to change over, I think.
Yeah, we could, do you have are we at the end of a move?
We're held here, but they've got a bit of tether to do some stuff.
Great. Now I'm gonna start turning over back here.
Okay. We'll just explore around until our full relief comes.
So for everyone that's just starting to tune in, we are currently going up an unnamed seamount located to the south part of Baker Island, part of the Pacific Remote Island Marine National Monument. Our team is getting ready to switch over to the next watch, so you might hear some technical conversations happening on the background very soon.
On talk on SPL, check, 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 check. So I've got audio on the quad, record 1, record 3. Then I'm going to check my metadata, 3 green, 2, 3, 4, 5, 6, all good. And then I can just check, make sure I still have exterior cameras there and there. Back to record 1 here. Oh, hold on. You just routed that to sat 3. We're going to put that back to Herc bubble. No big deal. Yeah, exactly. All right, so we're good. I'm just going to get set up with these guys. Pilots, both cameras are full wide, videos ready to roll.
Hey everybody,
that.
this is Kelly Farron. I'm taking over for Jashira in the communication seat. We're doing a little watch change right now at midnight, so that's why we're not doing too much exploring at the moment. So give us a few minutes while we get some things set up. We're excited to explore with y'all.
Yeah, they were both down there, weren't they? Right. Yeah, see if you end up staying in one spot and figure out. Yeah, because like it's going back that way. It's hard to say. I think it's correct like this. Like I was pointing that way and Harry on the right hand side of the screen. So yeah. So being a new thing, this switch is like yeah.
I can't make this pen go. I'm just gonna steal his.
Okay.
I just can't make it.
Oh, it's a secret master. That's so Argus pilots can't use it. Yeah, go ahead. They were at 55 to get here, but the next one, I don't know, we might have zoom out to see where.
So Mike, I think you've got a sample in forward port, Foxtrot, and Slurp 1.
Roger. So we don't need to go to waypoint longer. Yeah, the waypoints are just like they were dropped actually before we even got this. And that point 933.7 is just. That's just me dropping the target so I know where we're going, and then I use the Argus target bearing down there to get the full. Yep. Yeah, we got it. You'll have the keep track of the 1 kilometer thing. Yeah, you can use that as a it's just a goal, I think. That way they'll get done on a dive in 28 hours. They'll get to the top. If he flushes that. That thing is working well. Well below the limit, so. We'll zoom in there, video.
Holothorian.
Hey guys, Dwight's here in the back row.
Hey Dwight,
Morning.
are you at Sci 1 or Sci 2?
I'm at Sci 1 actually.
Sci 1, copy.
Sam's back here in Sci 2.
Sam moved over.
Different Sam.
Yeah, different Sam.
Oh, that's Sam.
Voice is a little different.
Yeah.
And Sarah's back here.
Sarah's now at Data.
Yep.
Cool, so here we are.
Yeah, I think
12
we
to
got
4
the whole
watch.
crew.
Yeah.
Right on.
All right, full wide. Coming out.
Late night watch.
It looked like there was a big coral.
So can you
Just
just
on the right.
update us, Nav, Albert? Has there been a ship move called, or are we holding station? Sorry, I can't hear you.
We have to move the ship if we want to keep going. Is everybody ready? Everything's ready.
Hang on. We're just getting set. Just trying to figure out what's going on. So, can you zoom out, HiPAC, to show us where the next waypoint is or what our general course is? Okay, so we're heading towards up the spine of this ridge. And I think as we go up, if we can kind of zigzag our way, you know.
So you want to do something like this? Oh wait, this is not the proper tool. So
Just stand by, Nav. I think we want to head up the spine, but while we're climbing up the spine, we want to look around to both sides, you know, so it's kind of zigzag our way, so we're not just doing a straight line and not looking around as we go.
yeah, sure. But we want to go up until here, like on a straight line, to get out this kind of.
Yeah, stay on the crest of the spine. Yeah.
And then once here.
When we get into that saddle, yeah, we'll look around some, and or we might just keep the ship moving if there's nothing that we want to sample or take a closer look at.
Okay, so that's 40 degrees bearing.
So I think they've been doing 30-meter moves. Is that right?
Yeah.
All right, you guys
So
ready for the first move then?
pilots,
Yeah.
are we okay?
Yeah, I'm good. Are you all right there? Yep,
Okay.
good.
Bridge nav. Can we get a step at 40 degrees bearing, please? 30 meters is good.
And then I think Sam saw something we wanted to take a closer look at as we start to get underway here.
Yeah, there was a bigger coral to the right of that sea cucumber. I'm not sure where we twisted to though.
So we were calling it steps, 30 meters.
Yeah.
And as they, if you keep calling it continuous for a long time, it's a full time.
Just make sure everybody's here.
Versus if you were to track a line,
This coral dead ahead.
probably calling it 20.
Yep.
All right.
So sometimes
Yeah, can we get a closer zoom on that?
Yep, stand by.
you'll figure out whatever it is.
Okay, video, zoom in there. Coming in. Holding there.
Can we get a polyp zoom?
Yeah, stand by. Any further?
Is that Paragorgia?
Oh, I have a ton of zoom left. Let me know
Looks
when you
like
want
a
it. Yeah, go on. Coming in.
Paragorgia, yeah. I don't
Any
know
little
the
bump,
species.
more will fly up.
Yeah.
Wow, a lot of brittle stars tangled up in
Paragorgia,
there.
it could be Myrcia, which is a Canthagorgia.
Okay, yeah, that's been like that all season. Yeah.
Yeah, there isn't a Canthagorgia that looks very similar to Paragorgia.
Can I get that bubble cam over the porch again?
It looks kind of like this. Sam, why is that kind of on the left? See how it's kind of floral looking, and then on the right, it's kind of out of view now.
Oh, on the right, it looks like all the polyps
Are they just
closed up and receded.
retracted?
Yeah,
Oh, okay.
so I don't know if we pushed some water current into it that it didn't like or what.
Yeah,
but
it's kind of cool to
It's
see
like
both.
two different kinds of coral almost. It's funny. It's like a bubble gum. All right, full
Coming
wide.
out.
Yeah, I'm not sure if it's in this guide.
Wide.
Roger. Okay, are we moving now?
I don't think so. Yeah, we
the
can.
ship is
Oh,
Argus should.
sorry.
Yeah.
Dwight.
So we've got a new crew here for all of you watching on this 12 to 4 watch. So maybe as we're making a move, we could start doing some of the introductions to get to know some of the people on the watch.
So the 30
Maybe we
meter
can
step
start
is
with the
all-
back row. We've got a few different scientists
30-meter
with
step
us
is
on this
almost
watch.
done.
So maybe Dwight, Sam, and Sarah, if you guys want to introduce yourselves quickly. Or not quickly.
How quickly should we introduce ourselves?
You can take as long
we're
as
out
you
of
want. You're the expedition lead, Dwight.
All right. I am Dwight Coleman. I'm a oceanographer from the University of Rhode Island Graduate School of Oceanography, marine geologist, and I'm sitting in the Science 2 seat for this watch, and I'm also the expedition leader, so my job is to kind of make sure that the expedition is going smooth, everyone's doing their job properly, and everyone's happy, and we're accomplishing all the science goals that we've set out to accomplish. So happy to be here with you guys.
Thanks, Dwight, our fearless leader.
All right, I'll go next. I'm Sam Georgian. I work for Marine Conservation Institute out of Seattle. And I'm back here on the science panel.
And I'm Sarah Seabrook. I am sitting in the data manager seat, and I am an oceanographer at Oregon State University. I'm finishing up my PhD there right now, studying many different habitats in the deep sea and shallow seas as well.
All right, thanks, guys. Happy to have you guys here. Do we want to do some introductions in the front row? Who do we have on nav?
Yeah. Hi, I'm Albert. I come from the University of Girona in the northeast part of Catalonia. I'm here sitting on the nav position for my first cruise this year.
Awesome.
Michael Hannaford, Herc pilot. Been out a lot this year. And yeah.
I'm Gabby Inglis. I'm Argus pilot. Been out a few times this year as well, from Rhode Island.
Awesome.
I'm Ed McNichol. I'm sitting on the video watch right now, but I'm going to be handing it over to Nick Reed shortly, so we'll be hearing from him soon.
Great. And I'm Kelly Farron. I'm sitting in the science communication seat. And thanks all for joining us tonight. We have a lot of people who are excited that we're back diving, and we are excited to be diving again too. So happy to have a lot of followers tonight. For those of you who may just be tuning in now, we are currently exploring a seamount located south of Baker Island, which is out in the Central Pacific. And we're pretty close to the Pacific Remote Islands Marine National Monument. And right now we're kind of following up a ridge of this seamount, looking for some deep sea coral and sponge communities as we explore down here.
Crinoid.
This has been a good dive for crinoids.
I'll have to look up what a rock pen looks like. I'm not really sure.
I'll zoom in there, Ed.
I'm not sure how many different species
Copy,
there are.
zooming.
The ones I've seen look kind of, have you seen that umbella sea pen? Look
Yeah.
almost like
Yeah,
that,
yeah.
but they're gonna be attached to
Holding.
a rock.
Okay. yeah, I've seen those. Other sea pens are usually in the sediment. Is that right?
All right, full wide. Coming out.
Yeah, sea pens are funny because they're one of the only corals we see down here that don't need hard substrate.
Right.
Yeah. Yeah. Really underdamped.
Yeah.
Do we know approximately how old this seamount is? We've got to know. Dwight, do you have any idea?
It's at least 100 million years old.
Ooh, okay.
Because of where its position is on the Pacific plate. We're in a pretty old part of the Pacific plate, so I believe this one was probably formed at a hot, well, it was definitely formed at a hot spot, but I'm not sure which one exactly, but a lot of these Phoenix Islands were formed at, I think, what was known as the Phoenix hotspot.
Gotcha.
So it's an ancient volcano, basically, that built itself up and erupted.
I think we're all right.
You know, over probably tens of millions of years, and probably the oldest part of it is something on the order of 100 million years old.
Wow. Cool.
Nav, what are the graduations between the lines, the bathymetry lines on the high pack? 10? Okay. Yeah, this isn't a small. It's 20 to 30 meters down, 25 meters down.
Yeah.
Wow, not many animals. I'm surprised.
Yeah, it's a little bare.
Yeah, when they first landed, it was really sedimented, but now it's pretty good outcrops.
Yeah.
Still bare.
We had a lot of those acorn worms down in the sediment at first, and then we ran across a grouping of tripod fish earlier.
Tripod fish? Tripod fish?
Yeah, tripod.
Cool.
Yeah.
I didn't see that.
When you get down into the saddle here, I will be about level with where you are now, I think.
Right, yeah.
So this is us leaping off that saddle then, right here?
Yes.
Wow, that's a leap. Or into the saddle, I guess.
I think it's
Yeah,
a
we're
little more
going-
gradual than it looks, but it's definitely still like a, if you look in Argus,
Yeah.
it's sort of
Yeah, I see
slope,
it.
but yeah, no, it's definitely, it's 25 meters down.
Yeah. And then back up.
Yeah.
I'm gonna mess with your color real quick.
Sure. If it's like a little bit of slope, yeah, if it's a little bit of slope like this, I'll probably stay here. But if it was any longer or any more severe, I'd probably spin the whole works of it around. Just go down backwards. You can't really see very well like this. Yeah, I try and stay as like close down this part of the frame as I can to help with the delta. Yeah.
Yeah.
Alrighty, thanks.
I think your tether's like 41 meters. I
Oh,
could be wrong.
jeez, that,
That
Yeah.
is long.
that does explain it.
Yep.
That explains why it feels long.
You think it feels long while you're diving? Wait till you're pulling in the recovery line.
Oh, really?
Yeah. You like get the milk crate half full, and then you get to the knot to the normal recovery line. You're like, oh, I got a long way to go.
Still going, oh gosh.
Yeah. Actually, it's not the recovery line, but the Jacob's ladder, the knot. I guess
Yeah.
it is
Daisy
recovery
chain.
line. Daisy
Should we
chain.
put another step? It's pretty far behind, but
Yeah.
it's like
Yeah.
following well.
Yeah, yeah. Another one.
Bridge nav. Can we do 30 meters at 60 degrees bearing, please? Thank you, Vitali.
Ship's moving slowly.
Yeah, it has like 1 knot of transfer speed, but
But we haven't been going 1 knot.
yeah, no, like definitely
Maybe
not.
0.2.
Yeah,
They're at 0.1 right now.
yeah.
Oh, really?
Yeah.
Yeah, max I've seen is 0.38, 0.4. Yeah, there's one thing that I've always noticed is that you have to put more speed than the one you actually want to move the ship. It's
Mhm.
like the
Okay.
I don't know like how is the control tuned, but it never reaches the desired speed.
Right.
Quick change in video.
So what?
Yeah, I definitely believe Ed about this
The tether.
tether length.
Yeah, it makes a lot of sense.
Yeah.
We were saying down in the
Yeah, that it
Missed
just
that it
looks
looked really
so
long.
far. Like it's a full 10 meters longer than what I was flying with earlier this year.
yeah.
That's a lot longer.
Herc sonar.
Yeah, I'll fix it. Albert, do you know how to check the orientation of the sonar?
What do you mean by check?
Because it goes back to defaults.
No. Operating that, I don't.
If we can get a zoom on that fish, that would be great.
Yep.
Sam, here's
Go
your
ahead,
fish
video.
ID moment.
I can confirm that as a fish.
Thank you. Confirmed.
Video, you zoom in there.
I was going to wait till he tells me. Oh, he did.
Wow, that's cool.
Partial.
Yeah, that's good.
Oh, it's one of those ones that is sideways.
Swim sideways.
Yeah. Do we want to zoom of the head, if possible?
Yes.
Hey, go in
Zoom
further
in
there,
tighter on the
video.
head if you can. Yeah.
Sorry. Yeah.
Nice.
Got a big old eye there. That's cool.
Yeah. Yeah. Hey, full wide, please.
Full wide eye.
Is that him?
Yep.
Possibly. Macroridae. Macroridae. Gadiform.
Something. No, I usually make a fool out of myself if I.
Squishy thing.
Yeah. Well, now the fish is trying for more action shots.
Mm-hmm. Want some more attention?
Some zoom there, video.
Yeah.
Yeah.
It's hotter than earlier this year.
I even brought like a second jacket, like
Yeah.
because last year it was so cold sometimes.
Sam, do you know what this is?
It's a stalked crinoid.
So is it a coral or not?
No.
Ah, you see, I was right to say I'm not sure if that's a coral.
They're more related to like starfish, right? They're echinoderms?
Yep.
Mm-hmm. What's that living
So
on
what
it?
kind
When you say more related, you mean
It looks like it has
in terms
a-
of like
A small crinoid on it.
What's that little thing above the yellow crinoid? Do you see that kind of off the rock?
This should be called sea
Oh, yeah.
palmetto.
A
like a
little
little, like,
sponge.
Sea palm.
Is that a sponge? Oh yeah. That's cool.
Okay, full wide, please.
Full wide eye.
I have an old professor who gets super excited whenever we see crinoids because she studies them in the fossil record from like, I don't know, hundreds of millions of years ago.
What are they like in the fossil record? Do they
I-
look a
I-
lot the same?
I think they preserve really well, which is a little surprising, given that they're just soft tissue for the most part. But yeah, I've seen some of the like her fossils, and like you can see like all the structural features, and it's really cool.
Okay, ship move is done.
Yeah, he
now-
got up to about half a knot on that
Yeah.
because we strung two together, I think.
Yeah.
Now it's
You'll do
now
that.
you're like almost 40 meters behind, or 20 something.
What's this up
Do
here?
you want to keep going, or
Sam?
Could we get a zoom on that coral in the middle?
Yep. Stand by a second.
yeah?
Okay, go ahead, video.
Looks like a bamboo coral. Oh.
So that is a coral.
Yes.
So do you say that that was a bamboo coral?
Not sure. I don't see the nodes that we typically would.
Oh, okay.
So actually, I'm not sure. Might be a primnoid, actually. Can we look at the base a little?
Yep. Something worked around here. Zoom in more there, video.
Touch the rock.
Yeah, and then can you just pan up it slowly?
Yep.
Yeah, it looks like a bamboo, but I don't see the typical like node structure that I would expect. So I'm guessing it's a primnoid.
Right. Okay, full wide.
Full wide eye.
Falling behind here.
Yep.
Calyptrophora, maybe?
What is that?
Oh, yeah.
Cucumber.
Is that a s-
Or is it a sponge?
Sponge?
Oh, it's a sponge. Cool. Quick zoom as we go by.
So Nav, has the strategy been to not let Argus get too laid back for these?
What do you mean?
You don't want to get Argus too far behind. Are you
Well-
trying to keep Argus from getting too far behind the ship? You want to keep it right under the ship?
When it gets steep, we want to.
Yeah, if it
Okay.
gets the, but like it shouldn't, like according
Okay,
to
full
the
wide.
bathymetry,
wide.
it shouldn't be very steep. But that doesn't mean that we find like a 10-meter step at some point.
Okay.
But I wouldn't expect a very vertical cliff, like 30-40 meters cliff, because
Right.
these are 10-meter resolution
Right.
contours. But I think we can expect something like, oh, all of a sudden we have 5-10 meters
Right.
outcrop or something like that.
Thanks.
But I think we can wait a bit to put the next move that
See it
Hercules.
coming in the Herc sonar there.
Yeah, the next one, about 30 meters away, no?
Yeah. Yeah.
Argus is getting to be underneath the stern of the ship.
Yeah.
Yeah, it's not back very far.
No, it's good.
It's not taking very long to catch up. That's kind of
Yeah.
nice.
Yeah, well, it's only 2,000 meters, so
Yeah.
the swing is not that bad. What I'm wondering is if Argus sonar is also like upside down, like Herc was.
No,
No, Argus.
Argus
looks
is
okay.
fine.
Okay. Ah, yeah. Well,
We've been seeing the sort of steeper,
Yeah.
brighter return, getting farther and farther behind, and now there's a new return showing up right in front, and that's the other side of the saddle.
Yeah, but it's like.
And you'll see it get closer. See that? The one we're heading towards is getting closer.
Yeah, but Argus is looking the other way around.
No, Argus is heading the exact same direction
We both
as
have the
Herc.
same heading now.
It didn't get updated here. Okay.
Oh,
Oh,
no,
yeah,
that's
no, that's
wrong.
right.
That's just to deceive
Yeah,
you.
yeah, yeah. Now I see it
yeah,
in
that's
your
a
compass.
total
It's like-
total trick.
Like, what?
Yeah. Yep, that's a total trick.
Oh my god. Okay, so you're in a better position now, so maybe we can put a new step in.
Yeah, go ahead.
Bridge nav. A step again, please. Thank you. Oh, there's a something there.
An enemy.
An enemy?
An enemy.
Yeah, I find this name extremely difficult to pronounce.
Yeah, I'd say. Yeah. Zoom in there, video.
Yeah, I always find like nice these like bright pointy ends that they have.
Yeah, that's really cool.
Yeah.
They must be really reflective in some way.
I wonder why. Like, what advantage does it bring you to have this kind of pointy thing? Like, not the pointy things, but like this kind of bright end at the
Yeah.
things.
I wonder what structure is there that's so reflective.
It's a good defense strategy for them to ward off other things from coming in, because most things recognize that as having maybe some toxins associated with it or something, or being something they want to normally stay away from.
So bright means no eat underwater.
All right.
A lot of times,
Full
yeah.
wide, please.
A lot of times bright means eat though too.
Yeah, yeah.
My biology is very basic, so don't get frustrated with me.
The currents calm
The
down.
sea star
Yeah,
up
something
there.
out of all that.
Yeah, you are straight aft now, like
Brittle star.
perfect position.
You don't usually see them just on the rock like that.
A little shrimp.
So at this point is where Dwight said that it's better if we can kind of explore the slope, like trying to zigzag to try to cover the maximum ground possible. But this looks steep here now.
Yeah. It is.
But it's cool. Is it, are these lava formations or?
Yes. Yep. I would say these are most definitely basaltic outcrops.
But we need to get some rock to confirm that, or not?
Yes, and we did pick up one already. So
We did? Cool.
we can keep an eye out for another rock sample to collect. It would be nice to get one with a biological specimen at the same time.
But that looks quite solid. That doesn't look like we can...
Yeah, on the steep spot, on the steep slopes, we probably won't find loose ones, but in the flatter sort of pockets where there's some sediment, we can find some loose ones, I think.
Yeah. So I was thinking now it's steeper, maybe we should put like 10 or 15 instead of 30.
Yeah, don't make a big move here. Argus is not very far away from it now, really.
Yeah, it's 25.
Yeah.
But are these rock samples going to be aged? Or?
They could be.
Also have a
you
40-meter
know, it's not-
tether now, so there's not as
it's
much need
expensive
to
to
get right
do
on
the-
top
Yeah,
of it.
you can
either.
let this one sit for a
uranium
second.
thorium dating or
Okay.
whatever they do. So
Because he's still got some
unless
up
the
to go.
scientist
Really
has
hot in
means
my
to do
sonar.
that.
Yeah.
It won't
It's
be.
starting to slack off
There's
now,
already
actually,
been
so-
age samples from
Yeah.
some of these seamounts, so I
Okay.
think they have a pretty good idea of the age.
The 40-meter tether is actually super deluxe for this. I've got
Good
like
for seamounts.
Super
got-
deluxe.
I've got so much room to work with. It's
Yeah.
really nice.
Yeah, it's nicer.
It's a super deluxe tether.
What is that?
Oh,
Yeah,
can we zoom
it
on that
really
small
is.
coral just at the bottom of the screen?
Oh yeah. Stand by.
Bathypathes, maybe?
Okay, video. Just go ahead and zoom in there.
Coming in.
Which one are we looking at? The top one or the bottom one?
Yeah, where the lasers are.
Ah, okay. I see.
It looks like the same thing that we saw earlier.
Here's hot.
That was identified by Tina as Bathypathes.
So may I ask something that might not be very smart? Like, I mean,
There's no dumb
All
questions.
questions are welcome.
it will be probably very basic, but like these things just filter the water to get food, no? But whose food are these organisms? Like, does somebody eat these corals or things that we observe here?
Yeah, there's some snails.
Snails.
Some of the sea spiders. Yeah, there's a lot of predatory snail species.
Okay.
You want a closer zoom, or are you okay with that?
No, I think that's okay. Thanks.
All right. Full wide.
Coming out.
You're still in good shape, Mike. There's nothing showing up in Argus sonar right now.
Roger.
Then shall we move?
Yeah. You can do a step.
We can do shorter than 30 though, if you
Yeah,
want.
don't do 30 anymore.
Bridge nav.
20 or something.
Can we step 20 meters, please? Thank you. Yeah, you can see it also in the screen that it kind of flattened out.
Yeah.
So Sam, we have a question about some of these corals. So since we're in pretty deep water, obviously this water temperature is pretty cold. How do these animals we see in these depths protect themselves from the water temperature? Do we know much about kind of the range of temperature they can live in?
Yeah, so corals, each taxa or species will typically have a often very narrow temperature range that it can tolerate. And for these guys, we do call them deep sea corals, but what they actually are is cold water corals. So they actually really like cold water like this for the most part. And you actually see a lot of stress, even just raising the temperature a few degrees with some of these guys.
It is 2.26 degrees right now.
That's cold.
Brr.
And how do you measure that? Like you said, like if you raise the temperature, it causes them stress. Like how do you measure that in a lab? I guess this stress is measured under lab conditions.
Lots of ways. You can measure baseline physiological parameters like respiration and feeding rate and things like that and see if that changes. You can also look at what genes they're expressing. So animals have heat shock proteins to help deal with temperature stress. So if they start churning out a lot of those, it's a sign that they're not too happy.
But you
corals
measure
a lot
that.
of times will just die.
You
which
measure
is a pretty
that
good sign that
within the. So you would have are these animals?
Yes.
So you would have one of these animals in the lab, or well, several of them. But then you would get chunks of it under different conditions. You would like kind of pinch them with a needle and extract something? You would like.
Yeah, so we, back in my graduate school days at Temple University, we had a bunch of corals that we grew in the lab. And we would, some of the experiments we did were temperature. So you have a control animal or animals, and you don't change the temperature, and you see how their respiration rate is. And then you have experimental group, and you increase the temperature and see if that changes.
But the respiration, you measure it like measuring how much oxygen is left in the water. You measure it by, because you can actually measure what the animal is respiration is, or how. I don't imagine how to measure that.
Yeah, so typically we put them in a really small chamber and we measure the oxygen in the water.
Okay.
This is one way to do it, and then over time they will draw the oxygen levels down.
Yeah.
Bridge nav, step again, please.
Big mound here.
Thank you.
Albert, those are great questions.
These are questions of somebody who knows almost nothing about that.
That's why they're so great. Our viewers are loving them, so it's good.
Somebody watch their mic.
Yeah. So yeah, this looks kind of the ridge, no? Because it was.
Yeah. You see it rise coming up again there.
Yeah.
It's just a little belly.
Not much living here.
No. Bad real estate.
Is that another stalked crinoid?
Yep.
They look so lonely out there like that.
I know. Looks like a lot of marine snow though, so there's must be plenty of food for these guys. Albert, you're still doing steps?
Yeah.
But only 10 or 15 meters at a time?
We were doing 30 earlier, like to get off the first kind of mountain, and now we went down to 20 to make sure we don't go too fast against the slope.
The steep, yeah.
There's also a sponge or something like that next to it. And also one in the bottom of the screen. No, once you look like closer to it, there are things almost, not everywhere, but there is life, like variety.
Want a quick zoom on any of these things?
I think we're okay.
Find something we haven't seen before.
Yeah, not impressed at all back there.
We will.
Scientists are hard to please.
We will try to find something new for you guys.
Boring.
Okay. Next step will have to be less than 60 degrees bearing, otherwise, like, because we've been moving this direction, and that looks more like this should be the direction.
Yeah, fix the bearing.
You've been asking for 040, haven't you?
No, 60. We were doing.
Just changed it.
Yeah, we're gonna do 35 now.
Roger.
Bridge nav. Can we do 25 meters at 35 degrees, please? Thank you.
So I don't know if you have the science chat up, but Tina just wrote in and said that the acorn worm we collected earlier is maybe in the genus Yoda. I don't know if we can add that to the sample notes.
Did you say genus Yoda?
That's what it says.
Like Star Wars Yoda?
Yeah, that's what I thought too. It looks
Spelled
similar.
the same way.
Yeah.
Yeah.